I regret having to disrupt any important discussions, but I think for the sake of time and because it's really the very last part of the very last session, we want to move on. And from the Horizon 2020 projects, we are still missing after the monogastrics and the ruminants, now the aquatic part. And the first speaker for the AQUA-FAANG project will be Dan McQueen. And I think Dan will tell a lot of stories about decoding the genome function to enhance how we can predict genotype from phenotypes.
Okay, hi everybody. And thanks very much for coming back after coffee to see the aquaculture talks. I thought I'd start by just asking the audience how many people are involved with aquaculture research other than speakers or panelists. Okay, this is good to see, thank you. Great. So I hope you can hear me okay. I'm Dan McQueen from the Roslin Institute in Edinburgh. I'm giving this talk also on behalf of Sigbjørn Lien at NMBU in Norway. Together we're co-coordinating the AQUA-FAANG project, which I'm going to talk around for a bit today. So just in the way of an overview of this talk, I'll give a bit of background and introduction to AQUA-FAANG, and then I'll talk about the data that we've produced and some ideas for how it can be used in support of precision breeding and genotype to phenotype applications, ultimately aiming for a more sustainable and profitable aquaculture industry. In AQUA-FAANG, we've focused on these 6 species shown on the slide, which represent the major finfish species in European aquaculture, several of which have much broader global relevance. So aquaculture benefits very broadly from genomics already, including in breeding programs. This figure on the bottom left shows the very famous example of the impact of marker-assisted selection for a major QTL region that imparts resistance to a viral disease, and the impact in the salmon industry of breeding that marker in, which has greatly reduced the number of deaths in the industry to make this disease much less problematic. And in that vein, GWAS and other methods have been used to identify a range of different QTLs. It's important to say in aquaculture, because of the species have a much more recent domestication history than poultry and livestock, there's more major QTLs still segregating that can be used in selection. But also many traits still, of course, have more complex polygenic architectures. And genomic selection's been a more recent introduction to aquaculture, but it's now operational in a number of species, including in industry in some cases. Of course, genomic selection is most effective when close relatives are being used due to the high linkage disequilibrium, but breaks down in more distant individuals, such as across generations. It's also important to say that in aquaculture, the big issue, the target for selective breeding is disease resistance due to the big impact that disease has on the sustainability across all different aquaculture species. Genomic selection for disease resistance then, because of linkage disequilibrium breaking down across generations, requires regular disease challenges. That's expensive. There's also growing animal welfare concerns about all those experiments. So including causal variants will, in theory, of course, improve the accuracy of genomic prediction when more distant relatives are involved, such as across generations, but there are few causal genes or very few variants known in aquaculture species. So it's well established in other species that causal genetic variants are enriched in functional parts of genomes where the genes reside, but also, more importantly, perhaps, or non-coding regions with regulatory elements that are influencing gene expression. The strongest enrichment of the top GWAS SNPs are typically overlapping things like promoters and enhancers, and also evolutionarily conserved genomic regions, which hasn't been mentioned specifically today. It's fair to say that historically the sort of data required to prioritize candidate genetic variants by their association with functional regions in the genome has been very limited for aquaculture species. And it's not a small undertaking to identify this sort of data, especially in non-coding regions like which harbor regulatory elements. So the AQUA-FAANG project, I've put here the main partners. There are 10 industry partners as well as 14 academic partners. This project's finishing very soon now, scarily soon in October, and it's been running since 2019. There's a website that has a lot more information if you are interested, resources and previous talks that give a broader introduction of things like the work packages and so forth, which I'm not going to go into detail today. But the main aim was to produce extensive functional genomics data in these 6 species and use that to annotate the genome and in effect pave the way for more precise breeding to improve disease resistance and other target commercial traits. And importantly, also to develop the competence and the methodologies in aquaculture species to actually expedite the uptake of genome annotation and its exploitation in the industry. And aligned with the sort of core philosophy of FAANG, there is an emphasis on data sharing, data standardization in AQUA-FAANG. In terms of the data that we've generated comprehensive genome-wide maps that capture regulatory elements, chromatin, epigenetic state, and gene expression, so not dissimilar to the previous talks, but probably not quite as detailed. This schematic on the right shows how you can use different sequencing assays that have been mentioned today, like ATAC-seq and ChIP-seq, particularly different histone modifications, to identify different chromatin states and identify things like promoters and enhancers in the genome. So in AQUA-FAANG we have 3 functional annotation maps, the dev maps and the body maps, which respectively represent a panel of tissues in embryos, not a panel of tissues, stages in embryos, and a tissue panel that's in both sexes, in both sexually mature and immature fish. So a relatively small panel of different tissues, but keep in mind we're working on 6 different species, so we couldn't go really, really deep in any one species. Kind of unique to AQUA-FAANG, I think. We also have what we call immunomaps, which is samples that in effect come from fish that have been challenged either with poly-IC or inactivated Vibrio bacteria, which respectively activates the conserved vertebrate antiviral response or sort of pro-inflammatory antibacterial response. This was done using the head kidney, which in fish is the primary hemopoietic organ where immune cells differentiate from stem cells, so very important for immune function. And that was done both in vivo and in vitro with leukocyte cultures from the head kidney. Without going at all into detail, this is just a table that captures the data produced across these 3 maps. This is not all the data that was produced in AQUA-FAANG by any means, but it is what I'm presenting today. So you can see the species in the rows and the different assays. And in total, we've produced around 4,700 sequencing data sets across the species. So I calculated that was about 50 trillion DNA base pairs of new information on genome functionality. And there's many partners and individuals involved in all this work, too many to acknowledge individually, but it's been an amazing effort to be part of. AQUA-FAANG has a dedicated work package for data analysis and sharing, led by Peter Harrison at EMBL EBI, and also with contributions from the Ensemble team. All the data in AQUA-FAANG from these maps I've presented today will be shared through the Ensemble genome browser, both to enhance the gene predictions, but also to produce novel regulatory annotations of the same type that Elisabetta introduced previously, with the final annotations due for release in October, but there's a lot of data that's already available on Ensemble if you're interested. And all the data and raw metadata describing the samples and how the assays were performed is available through the FAANG data portal for anyone to download. Also important to say, partners also performed their own data analysis across these different species. This was done using NF-core pipelines based on Nextflow. This was done to harmonize with the other Eurofang projects that were using NF-core pipelines, and also because it provided a way for us to standardize across the different species and experiments, so we had a more reproducible data. This is a very busy slide, but I wanted to show an example of the sort of data that we've produced. This is just data for Atlantic salmon. I draw your attention to the panel in the top right, and we have a similar prediction of chromatin states to what Gabrielle showed, but here this is for both the DevMap and BodyMap stages. You can see how you can use ATAC and the different histone chip-seq data to predict these different chromatin states like active promoters, active enhancers, I can't really see this, and the proportion of the genome that they overlap. The data on the bottom is just a data dimensionality reduction method called UMAP, which shows how we can take the open chromatin regions in the genome and look at the variation in how they're being deployed across different samples at different stages of ontogeny, such as in the embryo or in different tissues. Just to say we've captured a huge amount of biological variation in the data. That's all that's there to show. Maybe more relevant to this meeting, I wanted to hone in on a particular loci and just show how the data we've produced in AQUA-FAANG can be used to help prioritize functional genetic variation. This is just some screenshots I've pulled out from Ensemble for a very familiar gene, IGF-1, which encodes a hormone that's implicated very broadly in growth traits in a bunch of different farmed animals. Salmon have 2 copies of this gene. Salmonids have a duplicated genome, which means they have lots of copies of their genes compared to mammals. These are on different chromosomes. You can just see them highlighted. And then we can see things like the promoter predicted by Ensemble and the open chromatin regions from Ensemble. And these brown blocks are predicted enhancer regions in different tissues and different stages from the chrome-HMM prediction I showed on the prior slide. You can also see on here SNPs that have been generated by whole genome sequencing data. You can see just these 2 loci have hundreds of SNPs across them, and then I've highlighted the ones that are directly overlapping with regulatory elements. So you can go from having hundreds of SNPs to a smaller number of prioritized variants, and you can imagine how this would scale across the entire genome and all the genes. Just to talk a little bit around the immunomaps. So this effectively has captured regulatory elements and regions of the genome that are being activated or repressed in response to pathogen signals. This figure here is from the University of Aberdeen team. In the red, you can see regions surrounding very important antiviral genes in fish that are activated in response to the poly-IC, which is a viral mimic, as mentioned, such as H3K27 acetylation, which is marking activated regions in the genome. So this is probably the upregulation of the promoter, as well as maybe some proximal enhancers. And once again, we could look at these antiviral genes, and we can identify very clearly candidate regulatory elements distal to the genes or upstream of the genes that are being switched on specifically by these immune stimulations. And we can identify then variants within those regulatory elements that are being switched on when the fish are immune challenged. And we think variants overlapping these sorts of elements are likely to have a broad influence on disease resistance phenotypes, and this is something we'd like to look into more in the future. So the next talk from Sarah is going to talk a bit more around the dedicated work package that we have in AQUA-FAANG to explore more deeply the value of adding functional genomics data to selective breeding approaches. But just to try and sum up what I'm trying to say in my talk, AQUA-FAANG, in this schematic I've produced, has generated millions of new functional elements across these different fish genomes, including many novel regulatory elements. We can overlap those with genetic variants using tools, for example, in Ensemble like Variant Effect Predictor, where it's very easy to put a bespoke set of variants, for example, from industry in, and they can be overlapped with our annotations. In AQUA-FAANG, we've also produced several novel structural variation data sets in several of these species with the idea to give us more power to capture actual causal genetic variants. And then the sort of precision breeding applications that can follow are just things like updating SNP arrays with functionally enriched markers, more precise QTL mapping, and possibly selection on the functional or the candidate causal markers, genomic selection models of the type that have been discussed. Another point I wanted to make was genome editing. Having a more precise definition of targets could be very important, especially finding genetic variants that already exist in nature, where there's likely to be softer regulation around the practical use of those variants, possibly compared to novel edits. But there's always more work to be done, and having additional data, additional annotation will give more scope to identify causal variants. Some of the things we're doing already in the project have been mentioned already, like transcription factor binding sites in active regulatory elements, doing expression QTLs. There's some of that work in the project. And also, I think very important is conserved non-coding elements, which usually score very highly on their importance phenotypically. And in the future, there's other work that we could be doing. I've probably run out of time, and then are you being kind? Sorry, I'm nearly there. Just tell me to stop if I need to. This is my last slide, I think. Closing reflections. So AQUA-FAANG's created a large upstep in functional genomics data and competence. But I think it's very important to say a comprehensive understanding, exploration, and exploitation of this data is going to take years to reach fruition, especially when we think about how this sort of data can be used in industry. Partly, this is because when we take a broader global perspective, aquaculture, just a finfish, involves many highly diverse species, and the practical applications of even standard genetic and genomic tools is highly variable, from extremely advanced livestock level to none at all. So the realization of using functional genomics data on selective breeding is going to be gradual, and it's going to be different for different species. But it's important to say that the most advanced sectors, like salmonids, are already using AQUA-FAANG data to update SNP arrays in genomic selection models and in their gene editing aspirations. I'd like to thank the partners in AQUA-FAANG. Thank you for your attention, and I'll just leave that one up there to look at while people maybe clap, maybe.
Thank you, Dan. You're excused because you have more species than the others.
Very good.
So the next talk, which is already anticipated, is from Sarah Faggion, more specifically on the use of integrating these functional annotation data in genomic predictions. And we will do it the same as we had with the two other projects that we will have the final discussion afterwards. And then if you come closer, then you can answer the questions later on.
Okay, good afternoon. Thanks for being still here. So we will continue the journey into AQUA-FAANG with some results about the integration of functional annotation data in genomic prediction models for this particular disease in European sea bass, so a non-salmonid species, so a particular species. So just to provide a little bit of background about selective breeding in aquaculture, it's not so different compared to livestock. So it is mainly addressed to complex traits of economic importance, such as disease resistance, productivity traits, efficiency, and quality traits. But what is the problem with these traits? So to implement traditional selective breeding programs, we need routine individual phenotyping. But for this complex trait, it's quite difficult. So that's why we are interested in the implementation of genomic selection procedures. Another important feature of these traits is that they are polygenic, so they are controlled by many genes, and each gene has a very small effect. And the detection of these causative variants, so these genetic markers with small effect, is really difficult. What we know and what you have already seen in the other presentation is that they are mainly located in regulatory sequences, and they affect traits through changes in gene expression. Another important issue with these particular traits is that the animals to be predicted are not always related to the reference population. But what we need when we set up a selective breeding program aimed to improve these traits is a prediction equation that could perform properly even when the reference population and the candidate breeders are genetically distant. And this happens when we need to predict the genetic merit across generation or across populations. So what we can do to efficiently use the genotype to predict the phenotype? We can take advantage of functional annotation data, so we can filter the genetic markers according to their localization in functional sequences, and we can incorporate biological priors into prediction models. So you have already seen what is the main aim of the FAANG project, the general aim of the FAANG project. But what is really interesting is that we can use these resources to efficiently relate genotype, phenotype, and genetic merit, and then we can improve our prediction models. And this is really important because we can translate the research output into industry applications. So we can also facilitate the application of genomic selection in farms. So going in detail to our experimental design, the target species was European sea bass. It is a very important species for the Mediterranean aquaculture. And the trait of interest was resistance to viral nervous necrosis, or rotavirus. It is a major threat for sea bass hatcheries and farms. So that's why we are interested in this trait. And the main aim was obviously the integration of functional annotation data into genomic prediction models. So we produced an experimental population through a full factorial mating. So we mated 25 sires and 25 dams from a commercial population. About 1,000 juvenile fish were subjected to nodavirus experimental infection, and the challenge test lasted 29 days. The mortality was recorded twice a day, and for this presentation, the phenotype is the mortality as a binary trait. You can see bottom right, the Kaplan-Meier curve. So at the end, we had a mortality rate of 45%. Then the experimental fish were firstly genotyped with a madfish SNP array, so we obtained more or less 27,000 SNPs for 990 animals. But the parents and 40 experimental fish were whole genome sequenced. So in this way, we were able to impute to whole genome sequence all the experimental fish, obtaining 6 million SNPs per animal. And then we perform also the parentage assignment to reconstruct the pedigree of the fish. We performed the ADAC-seq analysis, so you have already seen this kind of analysis. These were aimed to detect active regulatory or functional elements in the genome. And for this particular analysis, we have two conditions, so the mock-infected fish and the nodavirus-infected fish. And for each condition, we sampled two tissues, so the brain, which is the target organ, the target tissue of nodavirus, and the head kidney, which is mainly related to immune response. So from the output of the ATEC-seq analysis, we detected the broad peaks and the narrow peaks. So we used the SNPs included in these broad peaks and the narrow peaks to pre-filtering the total number of SNPs. So we created different subsets to be used in genomic prediction, and you can see that we created different subsets according to the different combination of conditions and tissue. So for the broad peaks, brain mock-infected, head kidney mock-infected, brain nodavirus infected, head kidney nodavirus infected. The same for the narrow peaks. And we also consider all tissues and conditions all together for the broad peaks and the narrow peaks. In previous analysis, a major QTL was detected in linkage group 12. So this linkage group has been removed from the data set to avoid any interference of this major QTL, because we wanted to see if the functional data was actually useful to improve genomic prediction. in this table, you can see the total number of peaks that have been detected, the total number of SNPs before the removal of the linkage group 12, and the total number of SNPs that were actually used in genomic prediction. So the filtered genotypes were used to predict genomic estimated breeding values for the trait of interest through a single-step genomic plap. And the animals have been clustered according to the genomic relationship distance. So we tried to minimize the relatedness between clusters and maximize the relatedness within cluster. So we detected two main groups, and we performed a two-fold cross-validation. So each cluster was used as a testing set, one at a time. To assess the accuracy of the models, we compared the pre-filtered datasets, so according to the open chromatin regions, with a pre-filtered dataset where the genotypes were disrupted based on a permutation procedure. And the accuracy was computed as the square root of the square of the correlation between the estimated breeding values, so the breeding value obtained from pedigree-based BLAP and the GEBV, multiply the reliability of the EBV. So on the left, you can see the Pearson correlation between the EBV and the GEBV, which is very similar between groups, between data sets. And on the right, you can see the accuracy. We obtain an accuracy more or less of 0.34 for all the data sets. What you can see is that the data set where the genotypes were permuted, the accuracy is significantly lower. Then we we tried to understand the Pearson correlation between genomic estimated breeding value from the cross-validation and the genomic breeding value obtained using the whole data set, so no masked phenotypes. And you can see that the results are quite similar. So we obtain a Pearson correlation equal to 0.4 for all the data sets. And again, for the permuted data sets, the Pearson correlation is significantly lower. And then we wanted to see the performance of this genomic estimated breeding value in classifying the observed binary phenotypes. And in this case, we used different metrics. So the accuracy as true positive plus true negative divided the total number of observation, the area under the ROC curve, and the Matthews correlation coefficient. So these are the ROC curves for the broad peaks, so the data set obtained, including the SNP of the broad peaks for the different combination of tissues and conditions. And you can see that the accuracy, the area under the ROC curve, and the Matthews correlation coefficient are quite similar between data sets. And the accuracy is equal to 0.6, the area under the ROC curve is 0.6, and Matthews correlation coefficient 0.2. The same for the narrow peaks, so we obtain very similar results. So to wrap up, all the SNP data sets exploiting information on open chromatin regions were used in GBLAP with similar prediction accuracies. These accuracies are quite moderate. So what we can say is that prediction equations should perform properly, at least for some generations. But when genotypes were disrupted, prediction accuracies significantly decreased. And what we can say is that GBV are moderate classifier of the observed phenotype. But I want to stress that the validation procedure was quite stringent. So I can say that the results are promising. So they are first results, but what's next? There are other ideas. We have other ideas in mind. So we want to use other criteria to give a weight or to filter the genetic markers. So we have other data sets that have been generated through the AQUA-FAANG project. such as the ChipSAC data or the EQTLs. So we can use the EQTL p-values to give a weight or to filter the genetic markers. We can also use the chromatin status scores or other regulatory data. We can also use other approaches, such as Bayesian approaches for genomic prediction or using other softwares. And another idea is to convert the genotypes into images and try to use these images to classify the binary phenotype applying two-dimensional neural networks. So there is still some work to do, but we are happy to do it. So I want to thank all the people that have participated to the project, and thanks you for the attention.
Thank you for the presentation. I think we have time for 1 or 2 quick questions. So where are the interests in aquaculture, which raised their hand before?
Still Roman.
Right. Thank you, Sarah, for the most interesting talk of the whole conference.
Oh, thank you.
I actually have an actual question. What about linkage group 12? What
Yeah.
about, I guess you have ataxic results on this chromosome? Is that
Yes.
Is it correlated with the gene that you identified?
Yes, actually, it's not part of the work that I've been performed, but it has been performed by our colleague at the Roslin Institute. And what they found is that in this peak there is, it is associated with an interferon, which is, I mean, it has antiviral properties, so yeah, it's something interesting, but it's another part of the project that is not on this presentation.
And does it relate with the functional, like ataxic data?
Yes, it is overlapped, actually, yes.
Good.
I'm sorry, because we are running out of time, and I think everybody would like to finish in time for this session. So we have to come to the next project. Now we are switching to those projects which have not run for such a long time as the previous 3 ones. So we are only having 1 presentation for those projects which have been, I think, running for 1 year now, I think. So the first one will be on Geronimo, and it's about genome and epigenome-enabled breeding in monogastrics. So the floor is yours.
Thank you. Thank you very much, Christian. So I'm going to present you the Geronimo project, which started 2 years ago. So we are moving on chicken and pigs. So bye-bye, aqua fish. So. There are 2 parts in this presentation. One part on context and aims and work package organization of the project, and another part more focused on molecular aspect in relation to this session. So, as you know, we have to face big challenges as there are mice. Yeah. As human population grows, environmental degradation, food security, changes in social and cultural values. And in this context, the aim of Geronimo is to rethink animal breeding in pig and chicken, to move toward a more sustainable model, ensuring first production, but also promoting efficient resource use, animal resilience and welfare, and preserving genetic diversity. So a lot of aims. The strategy that we chose to do that was to use current omic technologies associated with multiple phenotypes related to production in terms of quantity or quality of products, feed efficiency, productive longevity, in particular in laying hens, fertility, resilience, and welfare. and this to contribute to try to improve knowledge on genome to phenome relationship, accounting for both genetics and non-genetics mechanisms controlling traits. So the Geronimo consortium is involved 21 partners from 11 countries with 3 private companies in pig and chicken, 3 professional associations as well in pig and chicken, experts in innovation and project management, and a lot of academic experts in different scientific fields. The Geronimo is organized around 8 work packages with 5 scientific work packages, 3 in blue here, in which all the omics data are generated, and this data has the fuel to the 2 other scientific projects in green here. In the P4, the aim is to develop methods based on genetics and epigenetics component of trait for improving selection strategy, like a lot of other projects. And in the P5, the aim, there are different aims, and one of them is to analyze the societal, ethical, and legal issues related to genomics innovations by using surveys. And another aim is to analyze the feasibility, the risks and benefits of genome editing by performing simulation studies, and also of epigenetic editing by an experimental pilot study conducted by Guillaume. I don't know if he's in the room. So let's go to the 3 work packages in blue, in which all the omic data have been generated. So as you know, the phenotype variation is determined by the gene expression regulation, which is under the control of genetic variants for one part, and for another part to the epigenetic modification, which are the result of environment. So based on this idea, the aim of the P1 and P2 is to identify biological mechanisms affecting trait variation, considering genetics and genetics and epigenetics component. So in the P1, we have quite large animal design to perform quite efficient GWAS analysis related to different scientific questions, for example, about sperm quality and growth and feed efficiency for pigs, and about laying rate, egg quality and feed efficiency during long careers over 90 weeks for laying hens. And here, just a few results about this P1. P1, you can see here a lot of QTL which have been located by different people for daily gain, back fat thickness in different breeds of pigs by using 20,000 genotyped pigs per breed, or about semen concentration by using 400 genotyped pigs, or in laying hens, for example, QTL for body fat reserve. And for all these QTL, which are plenty of good gene candidates. In the P2, we have smaller animal design compared to the P1. Yeah? And the aim is not to perform GWAS analysis, but just to compare epigenetics and genetics variant between different environments. And these environments are different transport and rearing conditions here in chicken only, between different diets or different ambient temperatures or seasons or different ages and generation for pig and chicken. So a lot of experiments in this work package led by Tatiana, which is here. who is here, sorry. In the double P3, one of the aim is to map genetic and epigenetic variation in different chicken and pig local breeds to propose strategies for optimizing the conservation of genetics and epigenetic diversities. So after 2 years, we have accumulated a large number of animals, of samples, of omics data, more than 80,000 genotypes and phenotypes in pigs and chicken, around 2,000 transcriptomics data, mainly poly-RNA-seq, but also small RNA-seq and ribodepleted RNA-seq for some samples. And coming soon, around 10,000 epigenomic data, so it's a methylome data, with associated epigenotypes. And with this omics data, we perform genetics and epigenetics analysis like GWAS, allele-specific expression, EWAS, like in another European projects, to try to decipher the part of genetics and epigenetics involved in the variation of phenotype. About the pipelines that we used in Geronimo, we use pipeline from NF-core, which ensure the reproducibility of the analysis when they are available, like NF-core RNA-seq. Otherwise, we use in-house pipelines combined with pipelines already approved by others, in particular other scientists in human. For example, TensorQTL from the Broad Institute for EQTL analysis, or FASER for allele-specific expression. About the genome assembly, we use the last version available on the website, Ensemble website, mentioned here for pig and chicken. And about the genome annotation, so the gene model in the genome, we use for pigs the version 1007 from Ensemble, and an improved version for chicken, combining gene models from different resources, including, of course, Ensemble, as shown just after. So I'm going to finish the presentation with some slides about this new improved version. So we successively gather 6 gene model resources. I don't have time to give some details about the methods, but these 6 resources were the 2 reference genome annotation, NCBI, WebSeq from NCBI, and Ensemble from MBL EBI, including gene switch RNA-seq data. The 2 resources related to the pilot AQUA-FAANG chicken multi-tissue RNA-seq data from FRH and the University of Davis, and 2 additional resources from INRAE and non-code, which is dedicated to non-encoding RNA. And using this strategy, we increased a lot the number of protein coding genes in blue here in NCBI or here in Ensemble to 24,000 in the new atlas, and the non-encoding RNA loci in red here to 44,000 loci. And we then tested the reliability of these gene models by quantifying their expression across 47 tissues. For that, we analyzed more than 1,000 RNA-seq samples from different data sets, well chosen to represent a good variety of physiological system in chickens. And we found more than as you can see here, more than 90% of protein-coded genes in blue here, and 80 of non-encoding RNA, which were expressed in at least one tissue, showing that the models of this new atlas are real. And finally, in order to easily work on this gene-enriched atlas, we created GIGA, a user-friendly tool, which is accessible on this URL, which gathers functional and genomics information. And for example, if you are interested in a QTL region, like in Geronimo, you can define this region quite easily, like here for the body fat reserve QTL on chromosome 2, and display very quickly all the genes present in this region. So here, for example, there are 17, thank you, 17 protein-coding genes and a lot of non-encoding genes. And you can then get various genomics and functional information related to these genes. For example, you can get the functional annotation like geo-ontologies with search here function, and select the genes with ontologies that are consistent with a trait of interest. And for example, here there are 2 genes, these ones, which are related to fat or lipid terms related to the QTL of interest. And you can also get expression across the 47 tissues with the possibility to, display graphical views, like you can see on the right, and for the 2 genes previously selected. And you can see here that it's possible, if you want, to prioritize, I don't like this verb, the genes expressed in a tissue that is consistent with a trait of interest. And in our example, CYP5A is highly expressed in the liver, which is a key organ of lipid synthesis, making these genes a good candidate for this QTL. And you can explore the region further by obtaining more information about, for example, genomic orientation between 2 neighboring genes, like protein-coded gene and a non-encoding gene, which can be a regulator of the protein-coding gene, or orthologous gene between mouse, human, and chicken, or co-expression, etc. So I would like, in conclusion, we think that this tool should be useful for the Geronimo project to explore the multiple QTL regions that we have located in chicken. And I would like to thank people who contributed to this atlas and this tool, and in particular my PhD student, Fabien, who is here, and Philippe Bardou, a colleague from INRAE. And it's too late to see the poster because it's removed. So, and thanks a lot for your attention.
Thank you, Sandrine. Are there questions for this Geronimo talk? If not, then I think we continue to the next species, coming back to ruminants. And the telling name is Rumigen. The presenter will be Sophie Matalia. I hope I expressed.
Yeah, Sophie Matalia.
Okay. So new breeding tools in the context of climate change, something which is very well present during the entire conference.
Yeah, hello everyone. It's a pleasure for me to present the Rumigen program dedicated to new breeding tools in the context of climate change, and I will focus on the first results that we obtained, that is the studies on heat tolerant traits and the study of the impact of heat stress on the performances of dairy cows at the population level. So this presentation was prepared with my colleagues of INRAE and IDEL from France, INIA and EAAP from Spain, and Wageningen University and Research in the Netherlands. So the aim of Remigen is to provide breeding tools, and mainly in dairy cattle, to improve the efficiency and the efficiency in the context of climate change, and to maintain the genetic diversity. And within this program, we have a lot of different competencies, so quantitative and associated to work packages, so quantitative genetics to study the heat tolerance traits and the management of genetic diversity. Epigenetics in order to better know how genome and epigenetics shape the phenotypes. So with the build of an epi chip, an epi chip that will be able to be used in on-farm, and some experiments. And the study of new breeding technologies, and more particularly genomic editing, in within species and between species. And this with the help of human social sciences, in order to evaluate the social acceptability of the new breeding technologies and of breeding strategies in general. So there are 18 partners of 9 countries, so I won't describe all of them. And as I said, now I will focus on one part of the subject, that is the measurement of the impact of heat stress on the performances of dairy cows, in order to define in a second time some traits related to that. For that, we used the existing large-scale national data. I mean, the phenotypes and the pedigrees that we use in the genetic evaluations, and the weather daily records associated to each herd. So in the first time, the studies were done within country separately, using the same approach, and this for production, health, and reproduction, so other health and reproduction. And then in the second time, we compared the results. So you may have seen some presentation in the session 55 on the traits that we obtained, and of a meta-analysis at the individual level during the interval meeting. So we have 5 populations, so 3 Holsteins in the Netherlands, in Spain, and in France, and 2 local breeds, Montbéliard in France and the MRY in the Netherlands. The performances that were used are the test day records on milk, fat, and protein yields, and somatic cell count, and the conception rate after the first AI. The period that was covered by this study varied between 5 and 11 years, depending on the country, and we had from 10,000 to 7 million of cows, depending on the population. So the daily weather information were provided by the national meteo agencies, and they were associated to each herd according to the zip code. We had different parameters, and finally we used the THI, which is a combination of temperature and humidity. So to study the effect of variation of THI on the performances, we studied each lactation separately, and we analyzed the performances using a model in which you have at each time the THI effect as a fixed effect, some other fixed effects to describe the environmental effects, the genetic value of the dam, of the cow, a perm and for production, and somatic cell count, a permanent effect since we have several performances per cow. So we tried several parameters, and finally for THI, the THI was defined as the average of THI within 3 days before the test day, so including the test day and the 2 days before for production and somatic cell count. And for conception rate, it was the 8 days after AI, so including the day of AI. So the effects, the other effects were defined by each country, so most of them are very common, but they are not associated by the same way. Yeah. And now I will present some results that we obtained in first lactation. At each time, green is France, orange is the Netherlands, and black is for Spain. The dotted lines correspond to the local breeds, and the solid lines for Holstein. And you see here for milk yield that at each time we have a break point and after a decline, but that this break point is not the same for all the countries, with a break point which is lower, much lower in France than in the Netherlands and than in Spain. And after you observe a decline of about 60 to 80 grams per THI in each country, and for the here a steeper decline that we could observe, of course, in Spain and not in the other countries, because we had more very high THI in Spain than in the other countries. So we have to wait for several years, and we will be able to estimate that even in France and in the Netherlands. So this temperature, so when we translate to temperatures, it corresponds to moderate and low temperatures. So beware that it's daily temperatures, including also the night. But however, it is much lower than heat waves that we recently had, for instance. So here, the situation for fat and protein yield, you see that we have about plateau and then a decrease. And again, the threshold is lower for France than for the other countries. And after, so we have a steeper decrease at the end, for and more marked for proteins than for fat yield. For somatic cell count, we have an increase of somatic cell count, which is negative, which leads to more mastitis, and which is more pronounced in France than with a threshold that is more pronounced for France than in the other countries. And finally, here you have the results for the conception rate, with decrease after a threshold which is more homogeneous between countries, between 61 and 64. And after, so when you see the slope, it's a big slope here, with about a loss of 1.3% of conception rate per point of THI for the Dutch and the French Holsteins. So to sum up, we observe a negative impact of increasing THI for all performances, which is more pronounced for production and for fertility. For instance, for France, an increase of THI of between 50 and 70 corresponded to 6 to 11% of the daily production, and the same increase corresponded to 10% of loss of conception rate for the Holstein in France and in the Netherlands. For production and somatic cell counts, we observed some differences in thresholds more between countries than between breeds. So this surprised us in the first time. But in fact, there are many different explanations, such as the farming conditions and the exposure to outdoor temperatures. So in France, a lot of farms so a lot of farming systems are based on pasture. So the cows are going outside and are exposed to these temperatures, which is not the case in most of the cows in Spain. And in the Dutch situation, the farms, so the cows that were selected for the study were cows coming from the biggest farms, so that probably are less exposed to outside temperatures. Another thing is the pasture also, because for France, in the particular case of France, where the moderate temperatures correspond to spring, which is a period of changing of systems, of feeding system, which have an impact on the performances, and that is probably confounded with the THI effect. So, and in Spain, they have a strong experience with high temperatures, so probably the barns are better equipped to mitigate heat stress. So all these things should be confirmed, but to do that we have to access to farm conditions such as systems, barns, or feeding information, and we don't have this information. In Spain, also, of course, we can assume that the cows are more acclimated to heat stress than in the other situations. For conception rate, the thresholds were much more homogeneous, but we observed, as I said, a steeper decline in the French and the Dutch Holsteins than in the French Montbéliard and the Spanish Holsteins. So for Spain, again, we can imagine that acclimatation and the better equipment may explain these results. And in Montbéliard, we can also assume, imagine that there are differences in genetic abilities. So to conclude, we observed a negative impact of increasing THI on all traits. So this is why it's very important to work on that and to include these traits in the breeding programs. We observed consistent patterns between breeds and countries, but with some variations of thresholds, particularly for production and somatic cell counts. But the low threshold, as I said, may be explained by things that are not related to heat stress, so we have to be careful with that. And this is why on the genetic studies, the thresholds that we kept, and so to define heat stress, are much higher than the threshold that we observed here. So in any way, traits on heat tolerance can be defined using combining performances and weather data. And as I said, the pattern of the curves depend on the traits. That means that the trait heat tolerance is very complex, and we should combine all this information to define what is a heat-tolerant cows. So I wanted to thank Remigen that financed this program, and thank you for your attention.
Thank you, Sophie. Is there a question regarding Remigen? So there's one.
Hi, Alexis Michenet, Hendrix Genetics. I'm not working on cattle anymore. Thank you, Sophie, for the very nice talk. Since there are no question, I was just wondering, is there something scheduled for the beef cows as well?
Not in this program, but I think that in the future we should build something. There were some presentation showing. So yeah, not in this program.
Odd question, sorry.
Thank you very much. Then we move on to the last presentation of an existing project, which is Juliaxis. I hope it's rightly. And
You pronounce it properly well, and I promise it's a real name.
okay.
Juliaxis.
I'm convinced about it. So it's about holoruminants and enlarging the number of the scope of large animals to really their small, well, co-living individuals.
Okay, thank you. Thank you very much, and thank you for being here still. And yes, I'm very happy to have the opportunity to talk a little bit about the holoruminant project, in particular because now we were talking about the other ruminant genomes that so far have been ignored in the European projects. And the presentation is divided in two main steps. The first one is a brief introduction to show where we are regarding the genetic basis of the ruminants, of the microbiomes, I'm sorry. And the second one is how we plan to contribute from our project. You probably note in the last decade that there is a growing interest in microbial-based intervention, not only in human, but also in plant and environmental science, and in particular in livestock as well. And we believe that the microbiome can contribute to the improvement of livestock sustainability by, for instance, by the reduction of the environmental impact, the discovery of new bioactive metabolite that perhaps can be one alternative to the use of antibiotics, and also to improve animal health and welfare. It is important to note that, this dependency between the host and the microbiome is not uniform across the animal tree of life, but in the top of the spectrum we have ruminants. Actually, the rumen microbiome is a good example of high fidelity and obligatory relationship of the host with the microbiome dating back 50 million years ago. And this is important from the evolutionary perspective also because some authors agree, and I agree also, that perhaps this large dependency create stronger links between the host and the microbiome as well. We know from human that the main drivers of the microbiota composition are diet and lifestyle. Diet and lifestyle. I'm just wondering how my microbiome is doing after the EAP. But the good news is that the ecosystem is resilient, meaning that it's able to go back to the normal state after perturbation, and it was just a short perturbation. And another good news here is that the host genetic also play a role. But let's say that a human is a terrible model for many things, including to understand host-microbial interaction, and due to many reasons, like domestication, partner of fidelity, and the reduction of environmental factors, livestock offer better opportunity to understand the host and genetic interaction. Still, if we check the literature, and it's something I did the last month, so if you have a paper on August, sorry, it's not included here, most of the effort in ruminants has been so far focused on nutritional strategies to modulate the microbiota. There are some studies looking for the genetic parameter of the microbiome, and there are also few studies trying to identify candidate gene from the host associated with the rumen microbial composition. From the literature, we know that the rumen microbiome is, to some extent, is heritable. Some taxons are heritable, and this is true not only for ruminant but also for monogastric. I know it was supposed to talk only about ruminant, but I think it's important also to highlight this here. And in particular, for the particular case of dairy cattle, we note about the existence of a heritable core microbiome, and the good thing, or the good news with that, is that in that article, the author showed that this heritable core microbiome is phylogenetically closely related, meaning that perhaps they are also functional related, cover one important proportion of the overall abundance of the rumen microbiome play a keystone role in the microbial network, and also is associated with some host trait, like for instance methane emission and some other trait like milk composition. We know also for beef cattle, another study later validate the heritable nature of some of these core microbiome, so it's not only found in dairy but also in beef cattle, and they also show that this core microbiome is not only at taxonomy level but also functional level, are heritable and genetically correlated with methane emission, and in one independent study also they show that it's associated with fatty acid composition in beef cattle. And as a genetician and microbiome enthusiast, we believe that they are really good news because we prove that the trait is heritable, is related with host trait, can be measured, so can be improved by animal breeding. Moving forward to the studies trying to identify host genetic marker associated with the microbiota composition. The first one is from Lee and co-workers. In that study, they basically use beef cattle, but later on, other studies arrive, not only in beef, but also in dairy cattle and in sheep. Because we are tired, I propose to check or to look in detail the paper, but using a very simple but easy to appreciate graphical representation. And here, each study represented as a node. The size of the study is proportional to the sample size, and the shape is related to the genetic background, either dairy cattle or beef cattle. And the node or the studies are connected if they are using the same taxonomy database. It's easy to see that most of them has been focused on rumen. That makes sense, but it's also easy to see that there is a lot of technical variability inside there, not only for the use of different databases, but also for the use of different bioinformatic pipelines. It's well known that we as research, we love biological diversity, but we hate technical heterogeneity, and this is a good example. We know that from the literature that all this technical heterogeneity first limit the inter-study comparability, and also is one issue regarding the replicability of the study. We also know how important is sample size, in particular for low heritable traits. This guy looked like me in the kickoff meeting and all annual meeting, asking for larger sample size to the partner, and Diego knows that pretty well. And this is the contest, and in the Holoruminant project, is a project that is coordinated by Diego Morgavi, who is there, and is one international effort, including not only partners from Europe, but also from Canada, the States, Australia, Cairo, Israel, and New Zealand. How we plan to contribute to that? Well, the main goal is to elucidate the role of ruminant-associated microbiomes and the interplay with the host. And we plan to do that by characterizing the microbiome using longitudinal studies to try to determine the connectivity from different body size, not only from the rumen, as well as better understand the genetic basis, relationship with host trait, and hopefully facilitate the adoption of the knowledge generated in the project by the then user. The project is structured in 7 work packages. I have no time, no energy to go to all of them, so I will be focused here mainly in work package 1 and work package 4. I just want to mention that work packet 2 and work packet 3 are the one in which most of the experiment has been done or are currently being done. And why work packet 1 and work packet 4? Well, because I think it's very important. The work packet 1, which is coordinated by Chris, is the work package focused mainly in the standardization of the methodologies and the consolidation of metataxonomics, world metagenomes, from the public database, but also the one that is being generated in the project. As output of this work package, we will be one open database, and the database will be open access and a repository with all the tools and the data generated in the project. Then in work package 4 is the work package for the analysis. We plan to integrate ecological modeling and genetic theory to better understand the association of the microbiome with the host. Is a work package led by Phil, and in particular in the task 4.4 is the one I involved together with ICSI and other partners of the project. We plan to identify microbial biomarker as well as run genome-wide association analysis and meta-analysis. So these tasks have two main steps. The first one is to analyze in almost 500 samples multi-omics data that will be generated at microbial level, including metaproteomic and metabolomic data, together with the metagenomic, obviously, and to see the association with host trait across life. And the second task is in the one that we plan to do GWAS and medium-scale meta-analysis, and for that we will use not only the sample that is being generated in the project, but also 1,000 additional samples from the Ruminomics project that was a previous European project as well. As expected outcomes, well, as previously mentioned, in the work package 1, we will release the HoloR database that contains all the information generated in the project, as well as public information, but standardized, and a public repository also with all the pipelines using in the project, together with microbial signature associated with host key traits, host genetic pleiotropic variants associated with host trait, but also with microbial signature. And I highlight this in red because two reasons. The first one, because I think it's a good opportunity here to collaborate with most of the project previous presented, like both REP and so on, and try to also score the SNP that we get from the genome-wide association analysis using functional information, and add also the information coming from the association from the microbiome. And also because I think that if we really want to propose that this marker be included in the genotypic arrays, it's important to be sure about the directionality of the allele. So if we find a particular SNP positive associated with any bacteria, for instance, we need to be sure that it's not the terminal for a trait of interest, because otherwise it will not be useful for the breeders, and hopefully contribute to the current state of the art regarding host microbiome interaction. And with that, I just would like to thank for the attention, and thank also to the member of the Holoruminant project. That's it.
Thank you, Eulaxis, for excellently staying in time. So this leaves time for some questions to the speaker. Everything's clear? Okay, there's one question.
Yes, Hélène Gilbert in RAI. Thanks for the presentation and the overview. I was curious to know, you mentioned that you have some key production index trait or something like that. Why did you choose to have an index of the production traits rather than target few different ones? And how do you think that it's more efficient than targeting a particular trait in which microbiota might have an effect, more direct maybe?
Well, I'm not think it's more efficient, first. And secondly, so in the project, there will be generated a lot of phenotypic information, and we just use the definition, key phenotype index to refer to all the phenotypic information that will be, it's not a index in the breeding genetics context. Sorry for that.
Fine, thanks.
You're welcome.
Thank you, Eulaxis. Now we're trying to get something new, which is the panel discussion. And for this, I would like to ask the 4 invited panelists to come up here to the podium. Okay. I think we invited on purpose 4 persons covering the different species which the major AQUA-FAANG project covered up to now, because we wanted to know from them what would be the expectations, what would be potential interests, and have a panel discussions between them maybe, but also between the scientists which presented the different projects, but maybe also with the auditorium. And before we start, I think it would be nice if the 4 panelists could briefly introduce themselves. So starting maybe with Abe.
That's me, Abe Huisman, working for Hendrix Genetics and lately as Director R&D for our swine breeding program, so overseeing our global breeding program for the swine species within Hendrix Genetics.
And then for covering the aquaculture, we have Romain.
Yes. Hello everyone, I'm Romain Morvezin, working at the CISAF, so a French breeding company association. I'm an aquaculture geneticist, working mostly with shellfish nowadays. But when I was hired at CISAF, it was for implementing genomics in aquaculture breeding programs.
Thanks a lot. And then I'm particularly interested what Olivier will have to tell, because it's about poultry, but also a little bit of biotech, so it's another field.
Yes, we have. So I'm Olivier Demer. I'm Deputy CEO and Chief Scientific Officer for Group Grimaud, and we have different species, including swine, poultry, and aquaculture through the shrimp. But we also have some biotechs, especially in fly genetics, on black soldier fly.
And maybe finally, last but not least, to keep the gender balance, but also the very important contribution from cattle. So it's Clotilde Patry.
Yeah, hello everyone. So my name is Clotilde Patry and I'm working for Alliance, which is the umbrella organization in cattle. And we cover, so we represent AI industry, but also milk recording organizations. And I'm also in charge of a society, a commercial society, that is dedicated to genomic services to breeders in cattle, sheep and goat.
Okay, thank you very much. You've all heard the presentations here from the already almost finished projects and some projects which are about to start or are starting and in the midterm. So what would be your perspective? What have you seen from, given your companies, the perspective for application? How could this integrate into breeding programs or into your also commercial effort, activities? Who would volunteer? So what perspective do you see from what you've...
I can start. No problem. So if we look at what we are doing in our commercial breeding program, we are in the business of selecting the next generation of animals. And we want to do that as accurately as possible. So if you look over time, then there's way more information coming available to include in that whole search for the most accurate breeding value. Well, genomics has been routine now for the last probably 10 years, but this kind of research will allow for more different data points that will allow us to do a better job at getting an accurate breeding value. Next to that, it will give us a better understanding of the whole genome. So maybe in future, when we will apply gene editing, this will also help us to open the black box a bit more, to know what we are doing, what we might be doing. And one other thing that I was thinking of when I was listening to all these talks, if we know more about this genome, we might also change our breeding strategies to come up with specific matings for specific end users, combining different genetic variants more precisely. So again, more accurately.
So it's about breeding and maybe also targeted matings, not in terms of genetic progress, but in mating the right animals to get the right end product. Others on the panel, do you have other maybe perspectives or other ideas?
Sure, I could have said what you have said. Actually, it's very true. But maybe I can add a concrete example. You've all seen the brilliant talk by Sarah, and about the VNN resistance, and you've seen that there is a major QTL on ALG12, so that was identified in AQUA-FAANG and in other projects, different population. this is a good example of a concrete example. There is already a fish born with this genomic information, so with this QTL, like resistance allele. So this is a direct outcome of a research program like that. So that's kind of traditional, like with detect a QTL, you have a resistance allele, and you genotype your fish in your breeding program, and then you can sell more resistant fish. So that's one example, but hopefully there will be others.
Mm-hmm.
So I can also give some examples. So of course, so first I would like to thank you all the speakers for the very interesting talk and presentation. It was not only easy, I would say even for me, to understand everything and to make the link with the application. There are still, I would say, some steps. But anyway, so I can notice that it will obviously participate to our better knowledge on the biology that is behind the farmed animals. And I think it's very important, not only for breeding, but also for all the management or all the services and support that we want to provide to the farmers. So just having this increased knowledge can help to have a better view on what is going on farms. So, and to advise in a better way the farmers. So that's also one point, and especially based on the studies on epigenetics, for example, the role of the environment, the role of the diet, and so on, it will help us a lot in the future.
So it's also pointing out that it's beyond breeding. It's beyond breeding. It's management and it's information for the farmers.
Yes, yes.
Maybe the biotech sector can also have some additional ideas. I don't know.
Yes, sure. So, of course, I join my colleagues on the comments and interest of all the presentation we had this afternoon. And to go further on Clotilde's comments on how we could use it behind only genetic selection on breeding, we see that when we deliver animals that we will be parent stocks of further offspring, which will be the commercial stocks, the environment will change the performance of the offspring depending on if they were produced locally or if the parents were shipped and then the animals were produced locally. And that makes a big difference in the performance at the farm level. So here it's not the breeding part, it's really how you provide the best product to your customers and the performance. And being able to understand how the methylation and how interaction might impact this and understand this helps you to understand from your genetic stock in your breeding farm to different locations for commercial applications, what would be the impacts and how to manage it. So it's also, as you stated, Clotilde, it's also behind breeding.
Thank you. Clotilde already mentioned that it was I really grant that it will be sometimes difficult to follow up the talks, but you might have also the idea that, okay, I see something, but there's a gap, there's a bottleneck, there is something which is missing. And now you have the chance also, we really intentionally plan that you can also ask some of the speakers, what didn't you understand? Where do you see a gap? Where do you see something, you have an idea, but you think research is still missing something where you would like to see something done. So it's your opportunity then also to ask the speakers.
Yes, of course. So each time that I'm listening to such kind of presentation, I'm always wondering what will be the weight of this new information in the final results on farm. So, how does it explain what is the part that is explained by this new information? And then, so for example, we were talking about a microbiota, for example. So it's very, very interesting. So we can feel that right now it's a strong lever that we can use. But what will be the cost also of getting this new data in comparison with what we will earn also at the farm level? So this is something that is missing to me when I'm listening to the different presentation. Of course, it's not an easy question, and it depends on context. But
we have 2 hours.
Yes.
I'm looking at the speakers who would volunteer to answer this question. I see a lot of people leaving the room. But this points, maybe this points towards that maybe for future direction of application, we should maybe include also experts with at least some knowledge in economy. So that it might take at least some kind of calculation about costs and benefits. Is this what?
Yeah, and it's also so we are talking always about data.
Yeah.
And the importance of data. And for example, at the genomic level, I think we have reached a level which is very, very, so we have a lot of genomic data. Then we need phenotypes, and as Sophie mentioned, so we need so more and more precise phenotypes and also precise information on environment. And I think this is costly right now to get this level of information, this level of details about environment and phenotypes. So that's so we have to raise the question of the cost of the data in comparison also with the progress that we can hope. But of course, there is some progress.
Okay, other concerns or other gaps or bottlenecks?
I think that we stated just earlier that the genomic selection is now routinely done and performed, but what made us able to do that, if we get back 20 years ago, we were looking at regions of the genome where we had what was called QTLs, and it was big regions with absolutely no information. And then there was marker-assisted selection. But what made the gap is that we were at some point able to have thousands of SNPs for a low price, quickly, and with a method which was very easy to implement to use it for routinely selection. And that's what we don't have today in the new phenotypes or informations we discussed today. But what struck me this afternoon, that for the first time, we were able, I saw for the first time for myself, at least, I don't know for you, the combination of many information for global evaluation. And I think for the past years, we've been a lot working on if we move this, we see this pathway moving. It was very, very global. And now we are able to have very precise data in large amount. And what lack for a global application in breeding is being able to have those for low price, quickly, and a method which aggregates all those information in a very simple manner. And we saw today
consistency.
Yeah, sure, of course. Yeah. And this is coming, I think, and we are in the middle of this. We went from pure science and information and knowledge, and we are getting to this aggregation and information and method that will allow us to use it for breeding. And that will make the difference, I think.
Yeah. So if I may add, I completely agree. In the aquaculture sector, the genomic revolution happened a bit later because it was more it was smaller and less well, there was less money in it, so the cost had to be down for it to be implemented. I think we will see the same kind of conundrum with the new technique, the new data that we collect, that it could be implemented in all sector only if it's cost effective for everybody. And I think we are all saying the same thing. The heart of the problem is the tool that we use. It was a SNP array, it could be something else now. And is it cost effective? How much information do we have? And how much more information do we have compared to what we had before? Because if we're spending thousands to get 2% more precise, maybe that's not worth it.
It's not going to happen.
Yeah, for sure. Yeah. That's obviously not going to happen.
So, yeah, I agree with both these all of them. Yeah. It needs to be
good panel.
We're all agreeing. That's not nice. I should say something else. No, but if we want to do a practical implementation, it needs to be cheap, it needs to be consistent, and it needs to be doable on large numbers of animals. Yeah. Otherwise, it's not applicable.
So maybe a little bit of a provocative question. So you're putting a lot of focus on being accuracy and particularly being cost efficient. I mean, that we have now genomic prediction is to a large extent due to the revolution we had on the genome sequencing, structure, variant, and so on side. At that time when this was done, the horizon to ever come to genomic prediction was rather, well, it was not very clear that this would ever happen, because only with the invention of SNP chips it became cost effective. Maybe a little bit provocative question. now you're only going for cost efficiency. Where do you see a little bit of a vision also on the scale of the knowledge where there are bottlenecks? I mean, cost efficiency is clear. When you invite people from industry, you always go for cost efficiency. That's clear. But what would be areas of visions where you could imagine, okay, there is something also on the information side which we might be interested in?
I may.
Yeah.
I may. I think there is still a gap of knowledge of the link between genotype to phenotype, and I think a lot of progress has been made in the past few years, but explaining a phenotype with genetic, genomic, transcriptomic data, it's still kind of fuzzy. But I believe, that's my opinion, that's also because we don't really know what a phenotype is and how to measure it. What's the good way to measure a phenotype and how to accurately... sometimes we want a phenotype because we think that's what we actually are breeding for, but it's often more complex than that, and I think we should emphasize more on the phenotyping methods and digging down on what's actually a phenotype. And to finish on that, I think also we mostly are doing genomics. We are taking animals, measuring phenotype, and looking at differences in genomes in a whole population. I think we could do the opposite that we are starting to do, is looking at genomes and producing animals with different genomes, genome characteristics, and looking at their phenotype, rather like an end point and not an entry point, if that makes sense.
Okay.
Clear.
Yeah.
Something that I would like to add also is that I distinguish the investment into the data for research from the fact that we want a tool that is cost effective. So that's the first point. And the second point is about catching also the diversity. So for example, in cattle, we are always, or almost always, talking about Holstein, or at least talking about Bos taurus. And what about all the diversity that is behind all the zebu in India, in Brazil, anywhere? So really, I would like maybe to see more studies that are crossing the information in first different kind of environments, but also different kinds of breeds and type of breeds.
Yeah, and if we go for the Green Deal with the emphasis on increasing biodiversity, and now with lots of tools relying on information being transmitted within population, it's definitely something, a frontier which we have to pass, going cross populations, going cross maybe even subspecies.
And it has been said in previous sessions that we need to have tools to for the local practices, so to be able to work on breeding, but in the local conditions. And I don't believe that we are able to do that with our knowledge on the Holstein breed. So, and also, for the future, we are more and more talking about crossbreeding, so which creates much more diversity also, or complex things to understand. So that's why it's even more important to understand what is behind all these complex traits at a global level before in our breeding schemes, to try to work with crossbreeding.
Okay. Maybe this touches also that currently we are almost exclusively thinking of additive genetic variants. Maybe we have to also, when we go for functional effects, functional information, then we can maybe also dig also in these other frontiers beyond simply additive genetic variation. I don't know how you feel about that. I mean, we have lots of colleagues here in the audience, so your chance to ask them, or the other way around, are there questions from the audience to our panelists? Oh, there's one in the back. Didier.
I probably speak too much during this meeting, but my feeling is that we are a little bit late in the race against the climate change. genetics is rather slow and we start rather late. And at least for cattle, for instance, nearly nothing is integrated in the breeding objective to prepare this adaptation. And we know in advance that the feeds will be very different, the heat will be there very severely, and probably more importantly, some pathogens from the south will emerge in temperate countries, and I'm quite sure that our animals will be highly sensitive to them, because when we send them in the south, we see exactly what happens. So, of course, we can cross with some genotypes from the south, but we cannot jeopardize all the genetic trend we have generated since 50 years. So we really have to think how to address the point of the global change.
Anything from the panelists? Is it more a statement than a question to the panelists, I think?
But what do we need to change? Do we need to change the animals, or do we need to change the environment in which we keep the animals? Because the talk on the total, was it temperature humidity index? The difference between Spain and France is a difference in the way they keep the animals, right? And in Spain they cope, and in France they have a problem.
Do we have to go more for G by E interactions?
Because that's the fastest way to change it. Genetics takes forever.
Yeah. I can add on the effect of climate change on the animal sector. In aquaculture, it's very, very true. For shellfish farming, for example, I don't know if you saw my talk yesterday, I was talking about oyster farming, and they are in direct contact with the environment and in direct contact with climate change. There is no bound, there is no way of putting them away. Mm-hmm. They are in the open sea, and right now, like 15 days ago, there were heat waves and all over the Mediterranean with huge clam mortality. So shellfish farmers are already feeling the effect of climate change, and indeed it's a rush, it's a race that we are losing, I agree, to adapt those species. I don't have any solution, I was just giving us an example, and a quite tragic one actually, because people are losing their farms.
Yes, for following on this question, my question to you is, what is your strategy in terms of genetic diversity to be ready for any event? So is it maintaining a set of lines, having crossbreeding strategies, having strategies to buy the competitor to increase the number of lines you have, or rely on systematic genome editing to create the diversity you have lost?
Go ahead.
All of the above. No, but what we're doing to maintain genetic diversity is we keep lines, and we keep them as large as possible, so we keep the variation within the lines as much as possible available. That's what we do. And then there's the other options you mentioned, buying the competition, gene editing. But what we do today is maintaining those lines at a proper size, large enough so that we're able to maintain that genetic diversity.
So of course we have the same type of strategy. Plus we also adapt the crosses to the different countries or regions, but like everybody, and that gets back to the remark about how do we manage at best the crosses and their impacts. How can we select with this anticipation of the impacts of the crosses? Because today we select pure breeds and then we have crossed products, and that makes big differences. But to get to the gene editing, I doubt we will have enough knowledge to know what to change just to adapt. Because gene editing is a method, but you need to know what to change to have an impact. And good luck with that.
Mm-hmm.
So I don't think it will be an answer for big changes.
Regarding adaptation in plant breeding, they try, although it's becoming difficult with Nagoya, but to incorporate tropical genotypes into their genotypes. And that it was always a big debate in plant breeding. In animal breeding, it was not because of the diseases associated with the livestock in the south. So the south gene flow to the north was diplomatic problems in plant, but not in animals. But if the environment is changing so much, do you think you could find some interesting genotypes in the south? And if so, how would you work with that or negotiate with the countries? Or
I would say that, again, so it means that first, before all, we need to have more knowledge about these tropical, for example, genes. Maybe this knowledge exists, so we especially need to enhance even more the collaboration to have a better knowledge on that kind of diversity before to be able to answer to the question, will it be a solution for us? So right now, except enhancing the collaboration and especially maybe the research collaboration with these other kinds of country, I don't see anything else. But we also know that we have some favorable conditions for production in the future. That even so, we have climate change. It will be maybe also worse and worse for us in Europe, but still we will maintain some good condition in comparison with other places in the world. So we need to discuss, I think, in between these genes and the location where we are able to grow animals or plants.
And a quick word from the aquaculture world. We have a very diverse sector, very diverse species possible, and very early in the breeding programs. Some breeding programs are only like 3 generations ago. So we still have plenty of diversity, so we hope that we can adapt. And if not, because some fish are cold water species, well, we also have fish that are warm water species. So some farmers are diversifying by just changing species. In France, for example, in oysters, there have been a succession of 3 different oyster species, and now we eat the Japanese one.
Thank you, and thanks the panelists, and also the. There's one more question. Yeah. Okay.
Thanks. I'm Ying from China Agricultural University. I focus on dairy cattle epigenomics. Actually, for AQUA-FAANG did a lot of work. My question is, in China, we did genotyping for genome. I think the cost is much lower now. That can be do a lot of population, a lot of samples. However, for epigenome, like DNA methylation or histone, something modification, that's cost a lot. For example, like 6,000 Chinese yuan for one sample for DNA methylome. So could you give us some suggestion how to solve or how to handle this problem? Because if we can get a lot of samples, epigenome data, I think it can be used for the genome selection or something like that. Thank you.
I don't have any solution to offer. Unfortunately, I encountered the same problem. We wanted to do some epigenetic work, and it just was too costly, so we didn't do it. But if you have a solution, please, if anyone in the room has a solution for this kind of thing, please raise.
But for epigenetics, we are still at a stage where it's not yet routinely used on the field, and maybe we are not yet at a stage where it's fully competitive. So of course it will be important for the future. So I believe that right now we have still the opportunity to share samples, share the data that we could be able to produce. And about epigenetics, there are some works that have been done and presented here. There are other projects. So for example, so Sophie, you talk about Rumigen. Within the Rumigen project, there are also some work about epigenetics. And one of the aim of the project among the different work package is to produce a chip, so the same technology as a SNP chip, to get a tool which should be able to produce data on methylation. And we hope, in fact, that at the end, if we need, in fact, a lot of samples, and we need also people to use this kind of tool to then reduce together the different costs. But so that's just a starting point.
I think for answering those questions, we're missing some stakeholder here. We should have genotyping tool manufacturer in the room, Illumina, Thermo Fisher, and all others, because I think it would be valuable to give them insight on what we expect and what they should be working on, because those are the people that will be solving the cost efficiency issues.
But maybe a little bit to wrap up the discussion, this is where also the basic research, for example, to pinpoint which variants to put on a chip, which would be the relevant diversity, which would be appropriate to put into maybe our lines there. I think basic research could really be a lever for practical application in our breeding programs. So there's this room for basic research. Nevertheless, I think the agreement was between panelists that what sometimes hinders application is cost efficiency. So maybe in the combination with basic research activities to try to be even more cost efficient than we have in the past would be beneficial for seamlessly integrate what we do in basic research to put into practical application. I think these are the expectations of what your stakeholders have for us. And there was one hand raised up, so to add a comment.
Yes, because behind the cost efficiency, we also mentioned the effectiveness and accuracy. And we saw this afternoon in the bovine project that when you combine EQTLs and information about the chromatin, information about QTLs and so on, depending on the model, you observe no improvement or even maybe sometimes a decrease in the accuracy, or you can have a strong increase by 30%. And that shows that we are still in the basic research, fundamental research of understanding how it works and what can make it efficient. And that's where we get from mass selection to GBLUP, and when you understand how it works. And then you can apply it without any stress about the impact on your program. And I think, again, we are at this stage we were 20 years ago on genomics. We reached this step, and that's a combination of cost effectiveness, but that's just a material aspect. But the fact is we need to understand first, and that we cannot do by ourselves. So that's where all we saw this afternoon is very precious, and thank you again.
Yeah, I think Didier has one more comment on question.
I have also maybe one comment in the meantime, just to say that we are talking a lot about cost efficiency, but for me it's also important to have this tight relationship in between industry and research, and we need this fundamental research to understand the biological mechanism, and it's interesting to have at least a relationship, which may be quite slight, but so that we continue to learn, to follow what is going on, and to also help into the discussion before biology and industry can target what is really, necessary on the tools, on the cost-efficient tool.
Yeah. Yes, I fully agree with that. It was part of my comment, but also the cost is really a question of quantities, of question of volume. Today is very expensive, but in 2010, a SNP chip, the cost of a SNP chip was €100, which is more or less the cost of any good technology now to obtain methylation, for instance. And if we demonstrate So we need quite a lot of money in research because the tools are expensive. But as soon as we demonstrate that it is efficient, therefore the market will develop, the volume will increase, and the cost will decrease. And there is no reason that we do not start to explore some ways just because of the cost. We have to find the money for that. And of course, maybe 90% of the test will be negative and will never go to application, but the rest will be efficient and cost-effective.
Okay, I think I'd like to wrap up finally then. I think in the end we came together indicating that in addition to cost efficiency, we definitely need basic research maybe to make things cost efficient, because this lies the foundation for cost efficiency, because knowledge is the basis for that. And I think our industry faces a lot of social, economic, environmental challenges, which I think we need to address. Our community is not really large, and we need to closely collaborate between stakeholders and our scientists to within the new maybe research infrastructure project we have in AQUA-FAANG to address your needs. And you can definitely bet that you will be approached by the research infrastructure several times on your opinions, on your suggestions, on your needs. And with this, I'd like to conclude the session. Thank you all for staying so long for the very last session of this long and exhausting conference. Thank you very much. Hope maybe we meet together this evening again for some more relaxed discussions. And wish you all a very safe trip home, and maybe hopefully see you again for one of the next research infrastructure, AQUA-FAANG infrastructure workshops or stakeholder meetings. Thank you all.
And thanks for the chairwoman.